than genetic risk factors alone. Now, often we intervene in these diseases only in adulthood once there are significant onset of symptoms. But we believe that by trying to understand how these early adverse events affect disease susceptibility, we can target intervention much earlier and therefore have a much greater impact in mitigating risk of disease across the lifespan. Now, in our lab, we approach this question from the perspective of hematopoiesis, and particularly from hematopoietic stem cells. As a brief introduction, hematopoietic stem cells are found in the bone marrow in both mouse and humans. It's a pretty well-conserved system. And they sit at the top of the hierarchy, where they're characterized, of course, by self-renewal capability, lifelong self-renewal capability, as well as multipotency. And so this single hematopoietic stem cell is capable of generating a very diverse panel of cells in the blood, from enucleated red blood cells and platelets and granulocyte macrophages of the innate immune system to B and T cells of the adaptive immune system. And hematopoietic stem cells accomplish the generation of this diverse panel of blood cells via a series of expansion and differentiation steps. And I just want to point out a few of the most important here. So the first step in differentiation from hematopoietic stem cell is the loss of self-renewal, but these cells maintain multipotency. And so we get what we refer to broadly as an MPP, or multipotent progenitor. The next most important step that I want to highlight this morning is the bifurcation of the myeloid and lymphoid lineages downstream of multipotent progenitor. So this is the first real specification step in hematopoiesis. And from here you get two very distinct lineages: myeloid lineages that again include red blood cells and platelets, but also cells of the innate immune system, and lymphoid cells that make up your adaptive immune system. Now, this is a very generalized schematic of adult hematopoiesis, but in my lab, we study developmental hematopoiesis, which is a much more dynamic process that occurs across multiple organs throughout development. And so the classic dogma of developmental hematopoiesis is that it occurs in waves of hematopoietic cell generation. And the first wave we refer to as primitive hematopoiesis. It initiates in the early embryonic yolk sac, and it generates a very limited subset of cells, primarily primitive red blood cells and primitive macrophages. And it's really thought that this initial primitive wave really just serves to meet the oxygenation and scavenging needs of the developing embryo. Slightly later in development, around mid-gestation, we get what's called the first wave of definitive hematopoietic stem cells that arise in the developing aorta, or aorta gonad mesonephros region. And these definitive hematopoietic stem cells are different from their earlier primitive counterparts in that they are now able to generate all mature blood lineages that are found in the adult. These definitive hematopoietic stem cells are also capable of being isolated at a single cell level from the very early embryo and transplanted into an irradiated or conditioned adult recipient. And this single cell from the mid-gestation embryo can reconstitute the entire blood system of the adult. And it is on the basis of this gold standard transplantation assay that we've typically thought of these definitive hematopoietic stem cells that arise in mid-gestation to be the precursor of adult HSCs. So these cells immediately upon leaving the developing aorta migrate to the fetal liver, which is the primary hematopoietic organ during development. And these definitive HSCs undergo massive expansion and proliferation. And right around birth, they seed the bone marrow, again, where they're thought to give rise to the adult HSC compartment. And we've tended to think in this dogmatic way, to think of this process as very linear. And in the same way, we've tended to think of immune development as very linear, where as these early HSCs mature, so does their immune cell output. Now, in the last decade or so, with the advance of more sophisticated lineage tracing and transplantation assays, we now understand that this schematic is far more complicated. So there have been a number of transient progenitors identified across development that give rise to specialized subsets of cells. And with the same types of approaches in the field of immunology, we now know that these waves of early hematopoietic cell generation also give rise to very specific subsets of immune cells during development. And we refer to this idea as layered immune development, where cells that are required for that particular developmental stage are generated from transient progenitors across development. Now, what's really interesting about these cells that come off of fetal precursors during development is that they are made only once and they go on to persist across the lifespan of the animal. And so these cells kind of come in two non-mutually exclusive and overlapping flavors. They include tissue-resident cells, like I've shown here, several subsets of tissue-resident macrophages, including glia, microglia, Langerhans, and alveolar macrophages. And they also include cells that we refer to as innate-like lymphocytes. And these are cells that are, excuse me, lymphocytes by conventional phenotypic definition, but they function in innate immunity. Now, these cells, as I mentioned, go on to persist across the lifespan in the animal. And what's also very interesting about them is that they're very poorly or not at all regenerated from adult bone marrow. So adult HSCs lack the capacity to generate these cell subsets. Because of that, these cells, which are often tissue resident, have a unique capability to self-renew and self-maintain as a population within specific tissues. They're also, as I alluded to, very functionally distinct from similar subsets of cells that come off of adult HSCs. So many of these tissues many of these cells seed tissues very early during development. And I like to think of them as being both educated by the tissue as it develops and also educating the tissue as it develops. So many of these cells play really important roles in tissue formation. Finally, the developmental mechanisms that specify these cells are really poorly understood. And part of my lab's work is just to understand at a very basic level what the cell of origin is for these cells and what are the mechanisms that specify very functionally distinct cells during development. But what I want to talk to you about today is a different angle, which is that many of these cells are dysregulated in diseases of tolerance. And so these cells are made very early in development. They populate tissues and help form tissues during development. And then at some point, they can become dysregulated and cause diseases of tolerance, such as asthma and autoimmune disease. So I want to start I want to tell you more about how we started approaching this question of how fetal hematopoiesis produces distinct immune cells by giving you a little bit of background of work that I did during my postdoc that led up to this idea. So during my postdoc, I used a lineage tracing model to define the contribution of fetal hematopoiesis to adult immunity. And in this model, which we affectionately refer to as the FLIC switch, we have FLIC2 promoter elements driving Cre recombinase across to the MTMG mouse, where you have a dual color reporter, a tomato stop, a GFP. And this FLIC2 is a receptor tyrosine kinase that is important in many different hematopoietic functions. It regulates survival, it regulates proliferation, and it regulates differentiation, particularly in the lymphoid lineage. For the purposes of this talk, the function of FLIC2 does not matter. So I'll say that right away. And for those of you that are unfamiliar with lineage tracing, the way this works is that all cells in these mice start out expressing tomato. When FLIC2 is expressed, it drives expression of Cre recombinase, and you get a complete genetic excision of the tomato coding region and an irreversible switch from tomato to GFP expression. And so because this is irreversible in a lineage tracer, what we get is all cells starting out red. Once a cell turns on FLIC2, it switches to green. And then we can map history of expression of FLIC2 because all cells that have turned on FLIC2 and all of their progeny will remain GFP positive regardless of FLIC2 expression. And so when I started this work, we had shown, surprisingly, that all adult HSCs express tomato. And we showed this both by phenotypic examination of the adult HSC compartment, but also functionally by transplantation, showing that only the red cells were capable of long-term multilineage reconstitution. Now, this is, as I mentioned, very surprising since FLIC2, which is also known as FLT3, regulates so many different hematopoietic processes. And the fact that all adult HSCs are tomato means that establishment of the adult HSC compartment does not at any time point during development involve FLT2 expression. And the reverse corollary of that is that any cell that expresses GFP during development cannot contribute to the adult HSC compartment because a GFP positive cell cannot give rise to a red cell. And so when I started these experiments, I went backwards into fetal and early development to look at the HSC compartment. And of course, we found red HSCs in the fetal liver that seeded the bone marrow right around birth. And we presume that at least some part of this red population gives rise to the adult HSC compartment. But we also, and we refer to these lifelong HSCs in our system as the adult HSCs. However, we also found during fetal liver hematopoiesis green HSCs. And we showed that these green HSCs were capable of seeding the bone marrow in the neonatal period, albeit at much lower levels. But as I showed you before, they failed to persist into adulthood. And we called these disappearing HSCs developmentally restricted HSCs or DRHSCs. Now, these DRHSCs are referred to as HSCs because although they don't persist into the adult bone marrow in situ, if we isolate them from the fetal liver and transplant them into an irradiated adult recipient, they can support long-term reconstitution of the adult blood stem cell compartment. We also showed that these DRHSCs were more proliferative, and so we suspect that they likely exhaust or are outcompeted once they hit the bone marrow in the neonate. And most importantly, we showed that these DRHSCs are functionally distinct from their tomato-positive adult counterparts. And specifically, they have a unique capacity to generate these innate-like lymphocytes that we know are only made during fetal development, like B1B cells and B gamma 3 positive T cells. So we went on to show that these DRHSCs have this capacity. They are specifically lymphoid biased. So on a per stem cell basis, they generate considerably more B and T cells as compared to myeloid cells. They're also lymphoid lineage primed. So at the HSC level, they are highly overexpressing critical lymphoid genes. And this is very unusual for an HSC. HSCs are typically thought of to be megakaryocyte erythroid biased, or at the very minimum myeloid biased. And finally, as I mentioned, we showed that these DRHSCs have a very unique capacity to generate innate-like lymphocytes, and these classic innate-like lymphocytes that we know have been shown in the literature to be fetal-derived. So coming from that work, the way that we have now conceptualized this as a platform to study what we refer to as a critical window of immune development. And what I mean by that is that we have identified a specific transient progenitor that we know disappears sometime in the postnatal period that is responsible for generating subsets of immune cells that persist across the lifespan. And we think of this now as a critical developmental window for establishment of the adult immune system. And a critical developmental window refers to the idea that the phenotype of the adult immune system can be shaped by intrinsic or extrinsic inputs during this critical period.
Anna, can I interrupt 1 second?
Yes, please.
Question from Tracy Lamb. The question is, did FLT2 expressing stem cells reappear under circumstances of perturbation in hematopoiesis in adults, such as inflammation or disease or cancer?
That's a great question, and we are currently pursuing that. We have some anecdotal evidence that mice that are sick in the colony end up with a GFP positive compartment, but we think that if they persist under homeostatic conditions without infection, that they are extremely low numbers, very difficult to detect.
Okay, thank you.
Thank you. Okay, so now we've chosen, so when I started my lab, we wanted to really test this critical window of development by applying an extrinsic input. Infection, and extrinsic input can be many different things. Infection during pregnancy, a maternal diet or nutritional status, or maternal exposures. And we wanted to test specifically how extrinsic inputs shape establishment of the HSC compartment during development, how this in turn shapes the function and output of these developmentally restricted immune cells, how this ultimately affects the makeup and function of the adult HSC compartment, and finally how all of these impinge upon immune function and risk for disease. And so when I started this work, and I should note that this is work that's being completed by two fantastic graduate students in my lab, April Apostle and Diego Lopez. April is unfortunately not joining us here in Utah. She's the only student that is staying behind, but Diego will be here next month. Before I start, because I don't know how broad the audience is, I just want to throw up a few examples of all the FACS plots that I'm going to be showing in the next slides. So we identify fetal semen progenitors by flow, and we first gate on cells that are negative for mature lineage markers and positive for c-KIT and SCA. And this compartment contains all multipotent HSCs and progenitors, so the HSCs and the MPPs and everything in between. Now, there are very many subsets of these cells that can be divided based on expression of multiple markers. We chose to use markers such as FLIC2 and SLAM family markers like CD150 and CD48. This is kind of a classic schematic for breakdown of the semen progenitor compartment. And I will go through this a little more in detail later in my talk. But I also wanted to point out that we do this with our flick switch model, where within the HSC compartment, we can differentiate adult HSCs and DR HSCs by tomato or GFP expression. And so when we started this work, we wanted to have a very simple system, and we borrowed from actually the neuroliterature that uses a low dose of poly-IC or LPS as a model for autism or schizophrenia. And so here we are injecting 20 mg per kg poly-IC into a pregnant flick switch mice at embryonic day 14-5. And we chose this time point because this is peak fetal liver hematopoiesis, where the fetal liver is almost entirely a hematopoietic organ, and HSCs that have seeded the fetal liver are undergoing maximum expansion. And so we inject this poly-IC at 20 mg per kg. It's a very mild intervention. Pups are born at normal numbers, normal size. And what we observed was that in the neonatal period, we saw a very specific expansion of our DR-HSC population, whereas our adult population was really unperturbed. When we looked into adulthood, we observed that this mild stimulation at embryonic day 14-5 resulted in the inappropriate persistence of these DR-HSCs into the adult period. So as I showed you before, whereas previously we do not observe green cells within a phenotypically defined adult population, under a poly-IC treatment during the prenatal period, we see a significant number of green cells present within the adult HSC compartment. Concomitant with this expansion of our DR-HSC, we also observe significant expansion of these DR-HSC-derived innate-like lymphocytes. So here I'm showing you B1A and B1B cells within the peritoneum, and similarly, our conventional B2 cells are unaffected. And we see the same pattern in the spleen, where innate-like lymphocytes in the marginal zone of the spleen are expanded, and follicular zone cells within the spleen are unperturbed. Finally, we also showed that these innate-like lymphocytes coming off of our expanded DR-HSC population are not only expanded at the cellular level, but they are also hyperactive. So on a per cell basis, these B1B cells are pumping out more natural antibody, including IgM and IgG3. And this is work done by a former tech of mine, Eric Liebisch. So we have this basic proof of principle where we now see that this very early perturbation is expanding our DR-HSC compartment and changing the function of the immune system by changing both the output and function of these innate-like lymphocytes. And so we wanted to step back and determine how this was happening during fetal development. And so we examined hematopoiesis 1 day after injection in the fetal period. And what we observed was a significant expansion of our stem and progenitor compartment. And this was quite significant, but also relatively short-lived overall. So by E17.5, this change is already dissipated. And we tracked this as a function of our different HSC populations. And what we observed was that our DRHSC was specifically sensitive, again, to our intervention, where we see a massive expansion of our DRHSC within a day of treatment, and our adult HSC is less affected. We looked at proliferation of these two populations as well, and what we observed was that our DRHSC was also more proliferative in response to our intervention as opposed to adult HSCs. So in general, the DRHSC is already more proliferative, but we see even fewer cells in G0, which is normal in fetal development. Most hematopoietic stem cells are proliferating. And when we track this across development, what we could see was that for both our adult HSC and our DRHSC, that our poly-IC treatment resulted in prevented these cells from a normal entry into quiescence. So these HSCs during early development are highly proliferative, but as they approach the bone marrow, they become increasingly quiescent, and that was completely prevented by our modeling of maternal infection with poly-IC. I forgot to mention what poly-IC was, I apologize. Poly-IC is a viral mimetic, it's an RNA molecule, and the approach was to mimic a mild maternal viral infection. So we next looked at cells downstream of the HSC compartment because we wanted to understand how expansion of our DRHSC was specifically affecting differentiation downstream. And here I'll say a little bit more about these different MPP subsets that I showed you broken down by different phenotypic markers. So in adulthood, you have a subset of MPPs with variable lineage bias, where your MPP2 and MPP3 pump out really your myeloid lineages, specifically MEG-E and GMs, granulocyte macrophages. And your MPP4 are your more lymphoid biased progenitors. And under homeostatic conditions, these lymphoid biased progenitors are more frequent. Now, there's been a lot of work done recently, within the last 5 years really, on how infection shapes and inflammation shapes adult hematopoiesis. And in the adult, what's interesting is that many different kinds of inflammatory stimuli, interferons, TLR agonists, IL-1, all have a very similar outcome, which is to bias the MPP compartment towards myeloid and MEG-E output. And this is done to, again, support rapid myeloid recovery in the context of adult infection. But I want to highlight that adult infection in almost every case incurs a myeloid bias, not only at the progenitor level, but now we know also at the HSC level. Now, when we examined these different subsets in our during the fetal period, immediately following our poly-IC injection, what we observed was actually, what we observed was actually that our lymphoid biased HSCs, MPPs, which are defined by FLIC2 expression shown here, were significantly expanded in our treatment condition. And when we again track this over fetal development, we can see that our MPP4, this lymphoid biased MPP, multipotent progenitor, is initially significantly expanded. And we do also see some expansion of our MPP3. But when we look at this as a proportion of cells within the stem and progenitor compartment, you can see this very clearly how expanded this MPP4 compartment is. And so this was a direct line of evidence for us to show that maternal infection likely evokes a lymphoid bias at the progenitor level. And we are now completing analysis of a single cell sequencing data set that suggests that these MPP4s are coming directly from our DRHSC expansion. So now we think we have an initial proof of principle that there is a critical window of development where extrinsic input like maternal infection immediately expands and specifically expands this DRHSC population by increasing proliferation. And this results in an expanded population postnatally that gives rise to an expanded population again of these innate-like lymphocyte progeny. And we also have shown that this DRHSC then inappropriately persists into adulthood. So now we understand that this very early intervention or immune perturbation can shape not only fetal hematopoiesis, but adult hematopoiesis and the trajectory of the adult immune system. And so we are now very interested in how this ultimately shapes risk for disease in many contexts. But I want to switch gears a little bit now, because most of the data I showed you were at this point really as a proof of principle with this poly-IC model, which is a very easy model to tackle, but not the best model for an actual human infection. And so when I arrived at and started my lab at UC Merced, I established this fantastic collaboration with Kirk Jensen, who's a parasitologist at UC Merced, to look at maternal infection with Toxoplasma gondii. Now, Toxoplasma gondii is an intracellular parasite. It's also referred to as a torch pathogen, which simply means that it is can be vertically transmitted from a pregnant mother to the child during prenatal development. It is a very common parasitic infection. It exists, its primary host is the cat, and it's excreted as oocysts in the feces, and can then expose humans in a number of different ways. You can eat undercooked livestock and be infected. This is the reason why women are told not to change their cat litter during pregnancy, because these oocysts are very viable in air, and they can be released from cat litter and cause acute infection. Now, as I mentioned, this is a very common and ubiquitous parasite. About a quarter of the US population has been or is chronically infected, but that's relatively low compared to some other countries. For example, in France, where they eat a lot of undercooked sheep meat, infection rates are as high as, chronic infection rates are as high as 90 to 95%. Now, vertical transmission is much more likely, or almost only happens during acute infection during pregnancy. So chronic infection in this system is not well studied. Finally, the reason that I love this model is that vertical transmission has been shown during pregnancy to be regulated by a very typified interferon gamma response. And we leverage that in our model. So we know that interferon gamma sits on this weigh scale where it regulates vertical transmission, but limiting vertical transmission occurs at the detriment of embryonic development. So the beautiful thing about the system is that we have access to strains of varying virulence. And so here I'm showing you two different strains, a strain we refer to as PRU, which is a type 2 parasite. It's much less virulent. And RH is a highly virulent parasite strain where you can have a lethal dose of up to 5. And so we use the system where we know that interferon gamma can regulate vertical transmission and also fetal growth. And we use it to ask how does the fetal hematopoietic system respond to an actual congenital infection? And we can do this with these different strains with or without vertical transmission. And we're also very interested in understanding how the fetus actually translates this immune response. So what does the fetus see from the maternal infection and the maternal immune response? What cells respond, and how does this affect fetal hematopoiesis and immune development? And so this is actually the thesis project of my student April Apostle, where she takes time-mated mice, flick switch mice, and infects them at embryonic day 10.5 so that we can get a solid infection by our critical period of hematopoietic development at E14.5. And in this case, all of our analysis is done during the fetal period. We are still working out the system to have these animals born live, cross-fostered, and survive without exposure to maternal antibodies. And so, as expected, a congenital infection generally has a very strong effect on fetal development. And so I'm showing you here total cellularity of the fetal liver, where under the most virulent conditions, we see an extreme drop in fetal liver cellularity. Surprisingly, this doesn't affect the stem and progenitor compartment. So here I'm showing you this KIT positive, lineage negative, SCA positive stem and progenitor compartment. And in our most virulent infection, this stem and progenitor compartment is highly expanded as compared to our control. When we look at HSCs, this is not being driven by expansion of our hematopoietic stem cells generally, but instead it's being driven again by expansion of this FLIC2 positive MPP4, which is our lymphoid biased multipotent progenitor. And so again, we see that this real maternal infection, this very beautiful system, also evokes lymphoid bias at the progenitor level. And so April has also looked at proliferation in these experiments, and we see that both our adult HSCs and our DR HSCs proliferate in response to infection. And this is not surprising. All HSCs across development and adulthood tend to proliferate in response to infection. Our MPPs broadly are not proliferating to the same extent. And so we think that this is really showing that our progenitor expansion, our FLIC2 positive lymphoid biased progenitor, is really being driven by the response of the HSCs to the infection and not by the MPPs specifically. And so because we think that this is really happening at the HSC level, April has now taken these red and green HSCs out of the fetal liver in the context of these infections, and she's performed competitive transplants with lethal irradiation. And we're now in the process of performing secondary transplants to look at how self-renewal and lineage output is changed in response to infection during development. And the data I'm going to show you today are from our primary transplants, as our secondary transplants are still being bled. And so this is looking at the proportion of recipients that show long-term multilineage reconstitution after transplantation of either 200, I believe this is 200 red HSCs or 200 green HSCs or GR HSCs from the fetal liver. And what you can see very clearly is that as our infection is more virulent, we lose self-renewal potential from our DR HSC. And when we look at the peripheral blood of these transplants, 16 weeks post-transplantation, we're looking at donor chimerism in our saline and our two different strains. We can observe that actually our DR HSCs are again falling off the charts. They lose self-renewal potential and they lose the ability to generate cells long-term in a transplantation assay. Whereas our red HSCs, our adult HSCs, have significantly higher output as compared to the saline-treated control.
Anna, I'm
yes.
going to interrupt you because
please interrupt me. I see a lot of questions.
It's more fun in the middle of kind of. So Dean Tanton asks, can you apply poly-IC or toxo earlier in development and see changes in yolk sac-derived tissue resident myeloid cells in pups? And then he goes on to say maybe those changes persist.
Yeah, no, that's a great question. We haven't done that at this point. We have focused mostly on these innate-like lymphocytes because we have this DRHSC that we can track as the cell of origin for those cells. But certainly this is a really interesting question that we're interested in pursuing.
So he also has a second question on there, which is, can you avoid need for fostering if give mom toxo early and then treat so it's cleared by 19.5?
Yeah, so we have, April has done some work where she's vaccinated the moms long before, so early, like just post-weaning. And that seems to be somewhat more effective. She has been able to get these pups to survive, but for a PhD, those were a lot longer experiments. And because no one has looked at maternal infection and fetal hematopoiesis, we decided to start with that.
Okay, and then one second question from
Sure.
Tracy Lamb. Is the toxoplasm phenotype dependent upon congenital infection, or is this transferred by the immune response of the mother?
It's a very good question. We aim to be able, on an individual embryo basis, to look at vertical transmission with these different strains. We haven't yet done that, but we know that for RH, there's almost always congenital infection, whereas for Pru, it's a lot more variable and less likely to have a congenital infection with the Pru.
Okay, and then Tracy asks, is there a way to infect the mom or mum with attenuated toxo that does not make it to infecting the fetus?
Right, that's a great question. And I'll show you some experiments where we're trying to look at just the maternal response and not whether in a context without infection, but looking at a similar question. I'll get to those data in a minute, but that's a good point. I mean, really, I'm less interested in the congenital infection and more interested in the fetal response to the maternal immune response. And so I'll get to some. We haven't done those experiments, Tracy, with an attenuated strain. But we think for the Pru in particular, that April has done some experiments where she's looked at whether these cells are, whether the fetuses were infected, and most of them are not. The other thing to note is that she has tested whether the HSCs themselves are infected, and they are not. So the HSCs themselves do not become infected by toxo.
Okay.
Okay, are there more?
No, that's it for now. Thank you.
Okay, great. It's better to answer questions as you're going along. So again, we observed that our tomato-positive adult HSCs, isolated from the fetal liver, have higher levels of chimerism after 16 weeks as compared to the saline controls. And when we look at this across multiple lineages, and I'm just showing you two here, one in the myeloid lineages, so these are granulocyte macrophages, and one of the lymphoid lineages, B cells, we see this across all lineages. So both our Pru infected, so our less virulent infection, and our highly virulent infection result in adult HSCs that have much greater output as compared to the controls. So these proliferating adult HSCs actually have increased self-renewal potential in response to maternal infection, which is again very counter to the data that comes out of infections in adult hematopoiesis. So as I mentioned, we're really interested in how the fetus translates the immune response. And one of the first things we did was to do a multiplex assay looking at various cytokines in both the amniotic fluid and the fetal liver. And in the amniotic fluid, we can see a very clear upregulation of interferon gamma. And this should not be surprising because, as I mentioned to you, the maternal response is a very robust interferon gamma response. But when we look in the fetal liver, we see something different. We no longer see upregulated interferon gamma. Now we're seeing a significant elevation of IL-1, both IL-1 alpha and IL-1 beta. And so this led us to try again to understand how the fetus is translating this. And one of the things we wanted to do was to compare the response of a congenital infection to a direct injection of the mom with interferon gamma. And we did these experiments both in wild-type mice or in flick-switch mice, but also in the context of deletion of interferon gamma receptor or interferon gamma itself in combinatorial matings that would make the mom or the pups deficient for interferon gamma. And so we use this model to ask, does the fetus respond directly to the maternal interferon gamma signaling, or is there some translation of interferon gamma signaling happening on the fetal side? And we also want to know how the direct response to interferon gamma compares to the response of an infection to tease apart if interferon gamma is the real driver of these changes. And so one experiment that April did that I thought was really cool and really basic is that she took interferon gamma knockout mice, so both the mom and the pups are completely deficient in interferon gamma, and she did a direct injection of interferon gamma into these pregnant mice and looked at what was found on the fetal side. And so both in the amniotic fluid and in the fetal liver, we could detect interferon gamma, suggesting that this maternal interferon gamma is capable of crossing the placenta, which you'd be surprised how little we know about what cytokines cross the placenta during development. There's virtually no information on this. So we found that interferon gamma was capable of crossing the placenta and entering the amniotic fluid, as well as the fetal liver microenvironment, and that these levels were almost comparable to the levels we see in the amniotic fluid and in the fetal liver during either a virulent Rh infection or a less virulent Pru infection. And so then, given this, we wanted to understand how the fetus responds directly to this maternal interferon gamma. And so we injected moms with interferon gamma at E14.5. So now we're switching from the infection model and looking at the direct response to maternal interferon gamma. And in this context, we're looking a day later at E15.5, and we see no changes to our adult HSC compartment. But again, our DRHSC compartment, so our green HSCs present in the fetal liver, are significantly impacted by this maternal interferon gamma injection. And similarly, when we look at proliferation, we see that our adult HSCs don't proliferate, but our DRHSCs, again, significantly proliferate, which accounts for this expansion. When we look at our multipotent progenitors, we don't see a whole lot of differences. This is again at E15.5. We might expect these MPP4s to expand a bit later. We see instead a significant decline in more myeloid biased multipotent progenitors. And this is accompanied by a lack of changes overall in proliferation in the MPP compartment. So again, this suggests to us that any changes downstream are really being driven by a direct response of HSCs to interferon gamma. And so when we wanted to test whether the HSCs themselves were specifically responsible. And so we performed experiments where we injected interferon gamma into combinatorial matings of our interferon gamma receptor knockout mice, so we could compare fetuses that have the interferon gamma receptor to fetuses that don't. And when we look at our stem and progenitor compartment, we see an overall expansion in response to interferon gamma, but in our mice that are deficient for the or knocked out for interferon gamma receptor, we still see this response of the stem and progenitor compartment. And this suggests that this response of the entire stem and progenitor compartment is not solely being driven by a direct response to interferon gamma. In contrast, when we look at the HSC compartment in the fetal liver, we can see also a significant expansion, like I just showed you, of the HSC compartment in response to a maternal injection with interferon gamma. But this change is significantly blunted when we knock out the interferon gamma receptor in HSCs. And so we think that this suggests, again, that our response to infection is mediated by a direct response of the HSC to maternal interferon gamma. There are some other responses within the multipotent progenitor compartment that are occurring independent of interferon gamma receptor signaling. And so this is what we are now seeking to tease apart in this system. So to summarize what I've shown you today, we believe that our initial work serves as a proof of principle to define a critical period for immune development. So we've shown that hematopoietic and immune development can be shaped by these different extrinsic inputs during fetal development. We've shown that the fetal immune system can respond directly to maternal inflammation during infection, and that this shapes not only the output of our HSCs, but also the function of these fetal-derived immune cells that come off of these transient progenitors during fetal development. We also have data that I didn't get to show you today, that this has long-term consequences for normal and abnormal immune function. So my other student, Diego, looks at how these early changes to stem and progenitors shape lung immune function and also susceptibility to asthma. Some of the unanswered questions, ongoing work in my lab, seeks to look at, again, how perturbation to the fetal hematopoietic compartment ultimately remodels immune output. We're using single cell sequencing and ATAC sequencing to understand how changes to hematopoietic output across the lifespan are enforced. We're very interested in whether these fetal-derived innate-like lymphocytes that are both expanded and hyperactivated, whether they retain something like innate or trained memory, like we now know that some early myeloid cells do. And finally, we're looking to translate this work into human hematopoietic and immune development. And this is one of the reasons I came to University of Utah. I just want to point out a few near future directions. So I come from a nutritional sciences department. My background is in folate metabolism, and I'm very interested in how maternal nutrition influences hematopoietic stem cell establishment. And I'm currently writing an R21 specifically to look at maternal folate status during development and how this shapes epigenetic regulation of HSC development and establishment. I'm really also interested, and this is very early stage, but very interested in how early perturbation shapes what we refer to as clonal hematopoiesis. And so clonal hematopoiesis has become a very hot thing in hematopoiesis, where we know that up to 20% of the aging population has expansion of very specific HSC clones. And this is associated not only with, as a precursor for many blood cancers, leukemias and lymphomas, but also for these diseases like cardiovascular disease that we now know are associated with aberrant inflammation. Now, what I'm particularly interested in from a fetal hematopoiesis context is the part that most people ignore, which is how does establishment of a very heterogeneous HSC compartment shape susceptibility to clonal hematopoiesis. And so I'm very interested in pursuing this question with the systems that we have. And so I think I'm good for time. I'd like to acknowledge my lab, in particular the people that have done this work, including, I can't see their faces over this thing, there we go, including Diego Lopez and April Apostle, who have really driven this work forward with help from former tech Eric Liebisch and some really outstanding undergrads like Lorena Moreno. I'd also like to thank, I'd like to thank also my collaborators, Luisa Arispe, who's actually now at Northwestern, University of Chicago, Chris Vollmer's lab, who does some single cell sequencing of our B1B cells at UC Santa Cruz, and Nicole Bongarth. And I'd also like to acknowledge my funding sources and take any additional questions. Thank you.
Okay, I'm going to interrupt and just get one question. Iqbal, I'm going to unmute everybody, but Iqbal, why don't you ask your question before you have to go?
Hi.
Oh, gosh.
That was a fantastic talk, Anna.
Thank you.
I'm at the University of Maryland, and just to go back to the original, the flick mouse, tomato and GFP, right? So how do you know that the lineages are not being respecified once the tomato is excised? Because more and more single-cell RNA-seq results indicate that these guys are surprisingly plastic and can go back to the hematopoietic cell. So I was just wondering, have you actually isolated these tomatoes and GFP expressing cells and did single-cell RNA-seq in a continuum population to determine whether this is truly what you call as, I call it the Dr. HSC, or you call it DR HSC.
I like that. So we haven't done single cell sequencing yet, although that's in the works. We have transplanted these cells, and they read out differently still. So they are functionally distinct upon transplantation and isolation from the adults.
But clearly, your results indicate that when you transplant them into an adult, they can, into an irradiated mouse, they can establish certain lineages, even though they are GFP positive.
Correct. And we think that's because of the conditioned environment. There's something about, I mean, this has been coming out actually with the advent of like barcoding lineage tracing, where you can track individual HSCs in situ. We know, we know that transplantation, output from transplantation is very different from what happens in situ. And that's why we say we call this cell an HSC, because by transplantation, it's capable of reconstitution, but there's something about an irradiated conditioned environment that permits that. That changes the self-renewal potential of those cells as compared to in situ. That being said, when you take this cell out, it still produces different cells than the red adult counterpart. So the transplantation assay is still capable of registering a difference between these two cell lineages.
Okay, thank you very much.
No problem.
Okay, one other question from Tracy. Tracy, do you want to speak or do you want me to read it? I'll just read it. What other cytokines across the placenta? You already stated that it's unclear which ones, and she asked, can you do a Luminex particularly for other type 1 inflammatory cytokines that are more likely to interfere with signaling pathways involved in hematopoiesis at steady state? Is there any that are known to be in the JAK-STAT pathway?
I mean, we don't know of the only one that I know that has been shown to cross the placenta in early stages. There's also quite a big difference between, there's maybe 2 or 3 mouse studies that have shown that IL-6 is capable of crossing the placenta, but at earlier stages of development. So by E16.5, E17.5, that crossover is prevented by a mature placenta.
Okay, I had a quick, when you do the infections, is there a consequence to the fetus as far as erythropoiesis is concerned?
Okay, I suddenly became muted for some reason.
Sorry about that.
Oh, that's okay. We haven't looked directly at erythropoiesis, but we just got back some sequencing data that suggests that erythropoiesis related genes, both platelet output and erythropoiesis, is significantly affected by the poly-IC. So we haven't, erythropoiesis is a lot harder to look at in this model, maybe not, less during fetal development, but later it's harder to look at because the red blood cells don't really express the reporter. But the nucleated more primitive red blood cells would. But we generally haven't really analyzed erythropoiesis in these models. We've really focused on these lymphocyte lineages.
Okay. Any other questions? Okay, great, Anna. Thank you so much for your talk.
Thank you.
Wonderful talk.
Thank you very much.
Look forward to having you here at Utah.
Yeah, me too. I look forward to actually coming in sometime.
Someday. Okay. All right. Thanks again, and we'll see everybody in a couple of weeks for the next talk by Carolyn Philpott.
Fantastic.
Okay. Bye-bye.
Bye.
Great job, Anna.
Thank you.
Outstanding.
Thank you very much, Dennis.
Enjoyed it. Take care.
Okay, you guys too. Bye-bye.